Let us begin. My plan was just to go through the review because the review summarizes the test, so I think that's the best, in your best interest. If you have questions as we go, just ask. So at the beginning, the discovery of DNA as the hereditary agent, and so the critical experiments that I would expect you to understand are listed here. Let's take a peek at them real quick. We don't need to reinvent the wheel here, but let's take a look at the major points. So the first one for Griffith, bless you. We were looking at how he used two different types of bacteria, and the bacteria were distinct because one was the so-called R or rough strain, and one was the so-called S or smooth strain. And the difference between these two strains, if you recall, was that the R strain was non-virulent, meaning it did not cause infection, and the smooth strain was virulent, which in this case, the bacteria was, do I not have it up there? Maybe I don't. The bacteria was Streptococcus pneumoniae, and this was used by him as well as by Avery, McLeod, and McCarty. So we'll use some of this information for both of those experiments. But Fred Griffith's deal in his experiment was to put the cells into a mouse and confirm that, in fact, yes, they are deadly. So the type S cells, which have a capsule, remember they're smooth because they have a capsule around them. The capsule is a polysaccharide that makes it hard for your immune system to kill the cells. So we would struggle with this too. It's not a mouse thing. Then he had the R cells. He injected those. Mice are fine. Killed this bacteria. Then he injected heat-killed S cells, which this is a practical way to destroy bacteria but still make them immunogenic, meaning that if we were to inject some dead bacteria into your body, you might mount an immune response to it, but you wouldn't be in any danger. And then the next time if you got infected by real living cells, now your immunity is ready for that. So common way to develop vaccines. And then the weird result. So all those, nothing surprising about 1, 2, and 3. The weird result is that he took R cells, which are not virulent. He took heat-killed S cells, which are not virulent. The mouse died. And then, even weirder than that, when they did their little mouse autopsy, there were living cells in the mouse that looked like the S cells, but the S cells were never put in there, right? Only dead S cells. And so the conclusion, this is really the kind of the crux of it all, is that the R cells had been transformed into S cells by something from the dead S cells. Something inside the dead S cells had actually genetically altered the R cells. Okay, and so his conclusion, a term he used, it's not in that diagram, was that the S cells had inside of them a transforming principle. So essentially what he was saying was there's something in there that is genetic and is changing the hereditary nature of the other cells, but he didn't know what it was. So this would have been about 1928. There it is. Okay, so that experiment's done. Then we look at the next major experiment in this series of experiments, and that would be Avery, McLeod, and McCarty. And these guys were back in New York, so Griffith was actually in England. And back in New York, these three scientists realized we can replicate his experiment, but without the mice, just to make it easier. He was interested in vaccination. So for him, the mouse was the whole deal. Like he wanted to see a living organism altered by this vaccine. They are looking at his experiment and saying, hey, we can figure out the genetic agent. Let's forget about the mice. And so what they did is they treated, actually, let me see, there's a better way. I remember the first time we did this, I didn't like these slides. I should just take it out. They treated the killed S cells, and they added those to living R cells, the same thing that Fred Griffith had done, but then they did 3 different treatments, plus in experiment 1, I'll put EXPT for experiment. In experiment 1, they added an enzyme called protease that will remove the proteins. So if the proteins were the hereditary agent and you remove them, then nothing should happen. But they're not the hereditary agent, and so when you add the protease and remove the proteins, the cells are still transformed. So that rules out protein as the hereditary agent. Experiment number 2, they then added an enzyme called RNase. Okay, the RNase enzyme removes RNA. So if RNA was the hereditary agent and you remove it, then, principle, you won't transform cells. But yet, the cells were still transformed. So experiment number 3, which was the kicker, they killed our cells, they killed S cells, the living R cells, and then they added an enzyme that degrades the DNA. And if this DNA is the hereditary agent and you degrade it, the cells should not be, let me say it again. If DNA is the hereditary agent and you degrade the DNA, nothing should happen to the cells. And we know that DNA is the hereditary agent. So when you degrade it, the cells were not transformed. So the conclusion of this was that DNA was the hereditary agent. Right? Nothing to do with mice or vaccines, but they were able to use his experiment to figure out which of the three candidates might be the real deal. Okay, so then we go to the third, should be the third on my list there, Hershey and Chase, yep. The third experiment, and that involved bacteriophage. Just put Hershey and Chase, H and C. And these guys employed 2 important radioisotopes. And this was a really pivotal experiment, so you'll want to know the difference between these isotopes and why they used them. So they grew bacteriophage. These are viruses that infect bacteria. There's a big creepy bacteriophage right there. They grew phage in media with one of 2 different radioactive isotopes. The first one was S35. Remember, S is sulfur, so it's sulfur 35. So this is a radioactive atom that emits radioactivity that you can measure. You can watch this, you can detect it, you can follow it. And so they grew it with that, and the S35, because sulfur is found in proteins, the proteins of these bacteriophage virus will become radioactive. And so the term people use is label, because now it has something that can be detected. So this labels proteins. Okay. Then they did a second experiment where they used P32. And P32, which is a radioactive isotope of phosphor, does not incorporate into proteins. Amino acids do not have phosphorus, but they do have, but DNA does. DNA has a phosphate group with every nucleotide. And so P32 labels the DNA. So now you have an experiment where you have radioactive DNA, let me rephrase it. You have phages with radioactive DNA, and then you have a second experiment where you have phages with radioactive proteins. And so they let the phage go through their life cycle. I don't really have a picture of the life cycle here, but they let the phage go through the life cycle where they infect bacteria, they inject their hereditary agent, and then they make new baby viruses. And they ask the question, did proteins get injected as a hereditary agent, or did DNA get injected as the hereditary agent? Because they can follow this radioactivity. And so the conclusion was the sulfur, which labeled the proteins, did not make it into phage. The word they used for the second generation of phage was progeny. So you use kind of weird terminology with viruses because they're not organisms, so we can't say offspring because they're not living offspring. So that meant the proteins were not the hereditary agent, but the radioactive DNA was transmitted to offspring. So the same conclusion can be made from both experiments, which is that DNA is the hereditary agent. Okay, so they showed it with two different types of experiments. Both of these experiments were done in New York, if you're interested for trivia's sake. All right, what's our next one? Erwin Chargaff, Watson, Crick, and Franklin. Okay, so Erwin Chargaff, here's his table. Chargaff's main contribution to this was something that in retrospect was called Chargaff's rules. And Chargaff's rules were based on observations he had made with multiple different organisms and different species. In each of them, he measured the amount of the 4 nucleotides. Excuse me. And what he found was a pattern. The pattern was not that every organism had 25% of each, but the pattern was that whatever percentage adenine they had a similar percentage of thymine. Whatever percentage of guanine, they had a similar percentage of cytosine. Again, it's not perfect, and the reason it's not perfect is that nucleotides are used for more than just DNA, right? If it was just DNA, it'd be a perfect ratio, but they're used for other things too. But the pattern that emerged was that the A to T ratio was approximately 1:1, and the G to C ratio was also approximately 1:1. Okay, that's what we call Chargaff's rules. Now, Chargaff did not know this meant that A's and T's were base pairing or G's and C's were base pairing. Sometimes people misinterpret that to mean, or misinterpret that as what Chargaff's rules mean. That's not what it meant. This was prior to understanding the double helix. So his rules were this ratio. Okay, the importance of that ratio, though, was that Watson and Crick were able to use data from, I can't see it, I don't know why our thing's cut off there. They were able to use data from Wilkins, Franklin, and Gosling, which was X-ray diffraction data. I guess I should put that on our list here. Oh wait, that's no man's land there. Put it down here. So Wilkins, Franklin, and Gosling was their graduate student. A lot of people don't include his name, unfortunately, when they're telling this story. But these three guys, well, Wilkins and Gosling were male, Franklin was female. These three folks used a type of technology called X-ray diffraction, which is what you see in that picture there, which is when you blast a substance, in this case DNA, with an X-ray, and then you look at how those X-rays scatter as they bounce off the nuclei in the molecule. And so these guys produced an X-ray diffraction photo of DNA. And Watson and Crick were able to use that X-ray diffraction photo to figure out that DNA was some type of helix, that DNA had certain dimensions, certain width, certain repeated length, and then they used Chargaff's rules to say, well, if DNA is a double helix, that means anywhere there's an A, there could be a T on the other strand. Anywhere there's a G, there could be a C on the other strand. And they deduced that hydrogen bonding between those sets of nucleotides would be the right width based on this photographic evidence. So the photograph gave them some of the shape and dimensions. Chargaff's rules gave them some of the chemistry. And they knew that DNA was the hereditary agent because of the experiments that led up to it. So Watson and Crick were able to use all of the existing data to predict, or propose, I should say, the double helix. And they did not do, as has been noted many times, a single experiment involved with DNA. They were scientists, they were doing experiments, but they were actually studying protein. The DNA thing was their little side hustle. And they're criticized often for not having done any of the experiments, but the fact is, they used existing available information to figure out something that nobody who was doing the experiments had concluded. So we can't take that away from them. What we can take away from this, which is one of the sad components of this, is that Rosalind Franklin, was deceased when the Nobel Prize was awarded. So Watson, Crick, and Wilkins ended up getting the Nobel Prize. but Franklin, who might have gotten the prize because she produced actually the exact photo that they used in their proposal. But many people speculate, she either would have gotten the prize but didn't because she was deceased, or would not have gotten the prize because she was a female and it was 1953. But we'll never know. We'll never know the answer to that question. But Wilkins ended up getting the prize anyway. He had done a lot of the groundwork too, so it wasn't like he was just a consolation winner. Okay, I think that takes care of that part of it. Then nature of DNA structure, replication of DNA. Okay, so the nature of DNA structure, as we know, a double-stranded helix. The two strands of the helix are held together by hydrogen bonds. Right? These hydrogen bonds are easy to break, which is the point. It doesn't make DNA an unstable molecule. It just means that DNA can be separated into single strands and put back together as a double-stranded molecule very easily. That's the point. That's why you want it to be hydrogen bonded. And then the other thing in between each nucleotide, in this case it's highlighted in yellow, those are the phosphodiester bonds. Somewhere it says that in here, doesn't it? Thought it did. No, maybe it doesn't. So the bonds between, I'll write that out just to be sure, which are highlighted in yellow, are phosphodiester bonds. And these are covalent bonds. So the hydrogen bonds we want to break apart easily, but the covalent bonds that hold one nucleotide to the next in each of the strands, we want those to be really solid. We don't want the DNA to just be falling apart all the time. Okay, let's see here.
Wait, so those bonds hold together, they're like single strand?
Yes.
Okay. Sorry.
That's okay, I'll say it this way. Connect nucleotides within a single strand. They produce the polymer of nucleotides. Okay, then I think our next topic to hit, the 5-3 and the anti-parallel, yeah. So each nucleotide also has polarity with respect to the double helix. And that polarity is designated by the numbers 5' and 3'. And those numbers have to do with the carbons on the molecule. So if we label the carbons going around the deoxyribose of a DNA nucleotide, we start 1' at the carbon that's attached to the base, then 2', 3', 4', 5'. The 3' carbon has one of the most important groups to this whole process of making a double-stranded molecule, and those are hydroxyl groups. Hydroxyl, very simple functional group, oxygen and hydrogen. It's found on the 3rd carbon. On the 5th carbon, you have your carbon there, and then it's attached to a phosphate. Okay, and so we would say that there's a 5' phosphate and a 3' carbon. And if we were to link this up into a larger molecule of DNA, as we see on the right, I won't try and replicate that in this short time, but that means that on one strand of the molecule where there's a 5' phosphate, if we keep going down to the other end, we'll find the 3' hydroxyl. The other strand, though, is going in the opposite direction. So at the bottom here, opposite of the 3' end, we have a 5' phosphate. If we go up opposite of the 5' phosphate over here, we have a 3' end. So DNA is often diagrammed just as straight lines, which would look like this. Whatever direction one strand is going, the other strand is going in the opposite direction. Sometimes people will put arrows there, especially if they're indicating that the DNA is growing or being synthesized, but oftentimes not. But the 5'3" is telling us which direction the DNA can be synthesized, and it's also just a convenient way for everyone to label DNA sequences the same way. They always write them as if they were going in a 5 to 3 direction. All right, I think that's good for that. And then now how about replication? All right, so replication, the main way we want to approach this is to think about the enzymes involved as well as some of the proteins like single strand binding proteins. So when DNA is made into a second strand, each time that happens, one of the strands from the previous double helix remains, in this case the red, and one is synthesized, in this case the blue. Okay, so that's what's meant by semi-conservative replication. In order to replicate DNA, you have to split the original parent helix. And when that parent helix is split, it serves as the template for the new strand, which means wherever there's a G, we're going to bring in a new C. Wherever there's a G, another C. Wherever there's an A, we'll bring in a T. Wherever there's a C, we'll bring in a G, and so forth. So by having one strand in a single-stranded state, it serves as a template for the enzyme that is building this second strand, all right? And we call the place where this is happening the replication fork, because if we looked at it in a larger image, it kind of looked like that, which looks like a fork, okay? When the replication of a double-stranded molecule begins, there is an origin, okay? The origin indicates the starting place for replication, and depending on whether we are talking about a small genome in an organism like bacteria or a large genome in organisms like mammals, like you, the main difference would be that in bacteria, I think there's a picture of this, yeah. In bacteria, which have circular chromosomes like this, there's a single origin of replication. So for bacteria, they start replication here, and they work in both directions until they reach the other side and they're finished. But DNA like ours is too large. Like this, nothing in this is drawn to scale. Like this should be, you know, many, many times larger than that. Well, actually, is that true? No, it's not necessarily true. One single chromosome would equal a bacterial chromosome, give or take. All right, forget I said that. Anyway, it doesn't matter. The point is, we have many, many chromosomes, right? Like we've got 46 of these chromosomes. Some organisms have even more than that. And so if we started at one spot on each chromosome, it would take too long, take forever. So instead, eukaryotes have multiple replication origins that happen up and down their linear chromosomes. So that's one big difference in how it starts. Okay, now let's take a look at the enzymes, which is a point of emphasis in our discussion. The first enzyme, I believe on your list, would be helicase. And helicase, as we know, separates the double strands. Okay, so in our drawing, it's the blue donut-shaped thing, and it's going towards the replication fork. It's separating the DNA into two pieces. All right, so helicase separating the strands of DNA. After helicase separates the strands, you have to have a way to keep the strands from going back together. So that's where the breaded shrimp comes in. The weirdest interpretation of what a single-stranded binding protein looks like I think I've ever seen, but I do like breaded shrimp, so why change the diagram? But the single-stranded binding proteins do exactly what their name says. They bind to the single strands, and it prevents them from reforming the helix. Otherwise, the strand would be separated and would zip right back up. Okay? Which in fact is what happens when you make RNA during transcription. You open the strand temporarily and then it zips back up. So that would happen here as well, but we don't want it to happen here because we're going to separate those two strands permanently and make copies. Okay, the other one, which is on that diagram, so I'll just go ahead and touch on it now, is DNA topoisomerase. And within this word, you can see isomer and topo. Topo is a reference to the knot structure of an object, the topology. Isomer is, you know, a different form of the same thing. And so basically what the name of this enzyme means, if we take it at its literal meaning, is that it is changing the form of the DNA. And so this, let's look at their definition. Travels slightly ahead and alleviates coiling caused by helicase. And the way that I usually say this is that it relieves supercoiling, which is the term people apply to this. And the supercoiling occurs because the fork is separating the strands, right? If you separate something that's coiled, those coils have to go somewhere. They don't just disappear, right? And so it released supercoiling upstream of the fork, which is a more common way to describe what is ahead of the fork, upstream. All right, which is our other one? I think I asked about both polymerases on the test. I do. Primase, polymerase 3, 1, ligase. Okay. All right, now when we get to the part where we actually have to make the second strand, actually, do I set this a different place? There we go. When we start to build the second strand, there's a problem, which is that we can't just make DNA from scratch starting on a single strand of DNA. DNA can only be synthesized if there is a 3' hydroxyl on which to start building. So you have to have at least a little bit of the structure be double-stranded. So when you start, DNA polymerase can't do anything with that. But if you had a little bit of a nucleic acid with a 3' hydroxyl at one end, now DNA polymerase can do something. It can add to that end. And so the enzyme that allows this to happen is an enzyme called primase. The reason primase can make that little strip of double-stranded nucleic acid and DNA polymerase cannot is that primase is not a DNA polymerase. Primase is an RNA polymerase. And so it's not even making DNA, it's making RNA. Okay? And RNA polymerases are not constrained by that shortcoming of DNA polymerases. They can build a second strand just from scratch. Okay? So this is an RNA polymerase that makes the short RNA primer. All right, once that RNA primer is made, then DNA polymerase can come on and start adding to that 3' end. All right, the primer just has to get the process going. Now there are two polymerases, they don't distinguish them there, but we will. So the first polymerase that does the majority of the work is called DNA polymerase 3, or some people call it POL3. So this synthesizes the majority of that second strand at the fork. It does most of the work. I wouldn't presume to know the percentage, but I would say it's well above 95% of the work. Okay, so this does most of the synthesis of the second strand when you're making your daughter helices. But it doesn't do everything. So there is one other enzyme that we're going to need, and that's DNA polymerase 1. So for polymerase 3, I'll just say makes most of new DNA. But DNA polymerase 1 solves a problem that DNA polymerase 3 can't do. Oh, here we go. I've got 3 in 1. The problem that DNA polymerase cannot do is DNA polymerase 3 cannot remove the RNA primers, which means you're going to end up with a product which has both DNA and RNA. If I make the RNA in red, you'll have synthesized DNA, and then you'll have your RNA primers left over. And remember, you can only synthesize in one direction. You can only synthesize 5' to 3'. I'll put that up here. Makes most of new DNA in 5' to 3' direction. Every polymerase that's ever existed synthesizes 5' to 3'. So the problem comes when you're synthesizing 2 strands of DNA as the fork opens up more and more. Actually, there was a better picture of this. Here we go. As the fork opens up more and more, on one strand you can just keep going because you're going in the 3' direction, but the other strand you're going away. And so you have to synthesize some, and then you have to start again, synthesize some more, start again, synthesize some more. You can't just synthesize backwards into the fork. And so those little fragments are called Okazaki fragments, and we have to get rid of the primers and replace them with DNA. Okay, and that's the job of DNA polymerase 1. DNA polymerase 3 can't do that. So polymerase 1 removes primers, which are RNA, and replaces them with DNA. All right, now we have one job left to do. We've removed the primers, we've replaced everything with DNA, but there still is a section between the primers that hasn't been, does it show? Here we go. That hasn't been connected. So even though the RNA is gone and it's replaced with DNA, there's still a missing covalent bond. All right, and that missing covalent bond has to be sealed, as they say, and the enzyme that does that is the last on our list, which is DNA ligase. So this connects Okazaki fragments with a covalent bond. And that finishes up the job. Okay, let's see, what's next? All right, so that's all of that, I think. Anything else? I'm not going to ask about the telomeres. Okay, bless you. I wonder if that'll be our last sneeze of the semester. It's exciting. All right, so there's one section. Let me do 12 first because that'll be more consistent with our topic here. I'll build on what we've just done. All right, any questions about that stuff before we change gears? All right, onward we go. Okay, so for chapter 12, difference between prokaryotes and eukaryotes, transcription, structure of genes, stages, processing, translation, blah, blah, blah. Okay, so let's zip through this. We've just done this, so we don't have to beat this dead horse too much because we just finished this topic. The main differences between eukaryotes and prokaryotes, there are two main differences. One, prokaryotes do not have a nucleus, which is what makes them prokaryotes, so everything is happening in the same place. Transcription, which produces RNA from a DNA template, translation, which produces protein from an RNA template, all of this is happening in the cytoplasm, or the cytosol. It's the only place it can happen. In the case of I guess it wasn't going to be our last sneeze. But that's true. In the case of eukaryotes, we have a different scenario because we have a nucleus, and our DNA is in the nucleus, so we have to both copy the DNA into RNA, and then we have to get it out of the nucleus. So two jobs. In order to accommodate that, in addition to making the RNA, the second main difference is that RNA is going to be processed. The events of processing are to add the cap and the tail and remove the introns. So we'll look at those here in a second. Okay, both are eukaryotes and both eukaryotes and prokaryotes have some similarities, actually lots of similarities. One is that their genes have a beginning and an end. And in different ways, the beginning of the gene is designated by something called a promoter, okay, that says it signals the beginning of transcription. Another way to think of the promoter is that RNA polymerase, which is going to make the copy of RNA, has to start somewhere. So that's where RNA polymerase binds to begin transcription. Then the terminator is where RNA polymerase stops transcription and dissociates from the gene. So in between there, it produces what is called the transcribed region, which is going to be used to encode a protein, okay? Don't have to worry about those guys. All right, let's look at the process now. In the production of RNA, we have three events, just like we do for production of proteins. We have initiation, we have elongation, and we have termination. Okay, initiation involves the RNA polymerase, which is in purple here, binding to the promoter of the gene, which is in blue. It's cropped from this, whoever set up the projector made it too small, but the promoter is in blue, and that's where RNA polymerase binds to DNA and starts to separate the two helices. Okay, so instead of having lots of different proteins doing lots of different things, in this case, we have one enzyme that's calling all the shots. So it binds the DNA and opens up that double helix to make the open complex. Now, it's going to be traveling down the gene in this direction. As it travels, it's going to be synthesizing a strand of RNA, which is in yellow. So this is being made as it goes. Each time a new nucleotide on the DNA is available to be used as a template, a nucleotide will be added to the RNA. So when there was a C, we added a G. When there was a G, we added a C. When there's another G, we add another C, and so forth. So that the yellow RNA is growing as the polymerase moves along. Okay? As it moves along, the DNA, which is opening at one end to allow more RNA to be made, is closing behind it. So that little bubble that's available for the template to be utilized by RNA polymerase is a moving product, right? As the RNA polymerase moves, the bubble moves with it, opening ahead and closing behind. All right, when it gets to the terminator, the RNA and the RNA polymerase are going to separate from the DNA. The DNA is all too happy just to wrap back up, base pair with itself, and then the process is over. Okay, so it binds, it makes the RNA, which is elongation, and then it terminates when it gets to that terminator and separates from the strand. All right, so that's for prokaryotes and eukaryotes, but eukaryotes do have some additional steps that prokaryotes do not, and that is what we call mRNA processing. In mRNA processing, we make an RNA, which is the length of our original RNA encoding region between the promoter and the terminator, but the difference is, in that region, we have some sections that are called introns that are going to be removed before we have our finished product. Okay, the introns stand for intervening regions. They're between things that are going to be kept. They're intervening. The exons, which are going to make it into the final product, are expressed regions. They're going to be expressed and used as a template to make a protein. So when this RNA is made initially, it's called a pre-mRNA. The first thing that's going to happen to it is something called this is out of order. Actually, yeah, there we go. It's something called a methyl G cap, or the long word would be 7-methylguanosine cap, but typically people say methyl G cap. But this is a G nucleotide. So if you look up the structure of a guanosine nucleotide, that's it. Okay, and then there are 3 phosphates, because when nucleotides are synthesized before becoming part of a polymer, they are nucleotide triphosphates. So this will have 3 phosphates instead of the normal phosphodiester bond. You're going to have 3 phosphates. That makes the 5' end look different for this processed RNA. So if you just had an RNA that had a 5' phosphate and a 3' hydroxyl, that's going to be degraded very quickly in the cell. But if you have one that has a methyl G cap, a G with some methyl groups, actually several methyl groups. I don't know if they just show one on there, but it's actually multiple methyl groups. That's going to be different. And we said the purpose of the methyl G cap and the purpose of the poly A tail, which will also be added, are for stability. So the RNA is going to last a lot longer if it has a cap and a tail. And the ribosome, which is the machine that makes the protein, will recognize RNAs that are meant to be translated because they have the cap and the tail. If we take those away, the ribosome doesn't know what to do. Okay, so that's part 1. And then the second thing that can happen is the addition of a poly A tail. Again, this is an abbreviation. It should be about 50 to 70 to 100 to 150 A's, so it's actually a huge long string of A's. That also, as I said, adds stability and helps the ribosome know this RNA should be translated. And then the third thing, which is shown up here, it's kind of a clumsy, there's Mr. Shearer. Kind of a clumsy drawing, but the removal of the exons. So if this was a pre-mRNA that didn't have the exons, or sorry, removal of the introns, I misspoke, that hadn't been processed yet, the final product is going to be much shorter. So when it's all said and done, our RNA will have removed the introns and kept to the exons, and you have a much shorter, polished product the ribosome knows what to do with. Okay, then we have to translate this into a protein, And the ribosome is going to be following the rules of the genetic code when it translates the protein, so that wherever we see in our DNA or RNA a 3-letter code like CUG or CCA or CAU, the ribosome knows which amino acid to add based on which 3-letter codon is present. Two notable ones, or two groups in here that are notable, are the start codon, AUG, and the stop codon. In this version of the genetic code, there are 3 stop codons. And when the ribosome encounters the start codon, it knows that's where to begin translation. When it encounters the stop, as you would guess, that's when the ribosome ceases to translate. Okay, and remember we have these intermediary RNAs that are not messenger RNA, but are necessary for this process, and they're called tRNAs. The tRNAs, which are kind of shaped like a T, but that's not where the name comes from. The name comes from transfer. These are going to escort the amino acids to the ribosome to be incorporated into the growing protein. Okay, so let's take a look at how those guys work. We talked about this enzyme. I think it's on my list. It's not on my list, but you're going to want to know it for sure. Well, it is part of translation. But the enzyme that is depicted here, does it not say? Oh, there it is. Aminoacyl tRNA synthetase. There's a hyphen in there I'm missing. Aminoacyl tRNA synthetase. And this enzyme is critical because it uses ATP energy to join amino acids to the correct tRNAs. Okay? Maybe I'll say it this way. It uses ATP to pair amino acids with their tRNAs. And there are 20 different aminoacyl tRNA synthetases. There's one for every amino acid, right? So if you are a glutamate amino acid, there is a tRNA synthetase whose whole job, their whole reason for existing, is to attach you to the correct tRNAs that you belong with, okay? All right, so there's that. Then the ribosomes, think about their structure. There's a large and a small subunit. It's a little bit different in size for eukaryotes and prokaryotes, but the similarity is that there are tons of proteins, 55 and 82 respectively, and then the bacterial have 3 ribosomal RNAs, which make up a large part of the structure. Eukaryotes have 4. Okay, this is what the ribosome looks like. I would be comfortable with this structure if I were you, and you wanted to do well. So you would want to remember that there are 3 locations inside the ribosomes that each have a name: the E site, the P site, and the A site. Those 3 locations are critical for the function of the ribosome. Excuse me. We'll see how that works here when we go through the steps. Just like transcription, we have initiation, elongation, and termination. And when initiation happens, that's when the ribosome begins the process, which means it locates the start codon, and then it assembles its large and small subunit. The way that it locates the start codon, let's see, is it? Yeah, here it is. The way it finds the place to begin during initiation is that because the ribosome is made of both proteins and RNAs, there's an RNA part of the ribosome, in red here, or orange, that base pairs with a part of the messenger RNA. And so base pairing is a very specific interaction. So by doing that, the ribosome positions itself in the correct spot to get started. And the correct spot will put the start codon in the P site of the ribosome. Okay? Once that start site is in the correct location of the ribosome, the methionine tRNA, which is our start codon, tRNA, right, AUG, is going to interact with the start codon, and then the large subunit is going to land on top of it, and now the ribosome is ready to go. Sometimes, I think they call this the pre-initiation or initiation complex. There you go. So this ribosome is ready to go because new tRNAs can come in, and then they'll make the peptide bond and then keep chugging on in the 3' direction. All right, so the second stage then, which is elongation, is where we already have a tRNA in the P site. New tRNAs come into the A site. The existing polypeptide chain is transferred to the new amino acid. And then this is, if you follow the pattern of the blue, where the blue codon is there, the ribosome has moved 3 nucleotides to the right. And when it did that, this tRNA got bumped into the E site, which is the exit site, and it's on its way. This tRNA, which was in the A site, is now in the middle site, which is P, which means the A site is now open, which means we can bring in a new friend. And so this continues until we get to the stop codon. So moving 3 nucleotides at a time, bumping up tRNAs out one end, bringing tRNAs in the other, and building our little polypeptide. A question I know I will ask you is this: peptidyl transfer reaction, a bond forms between the peptide chain and the amino acid, in other words, formation of the peptide bond, and it is catalyzed by, do you remember? What catalyzes the formation of the peptide bond? Is it a protein or is it an RNA?
A protein.
That's what you want to believe.
It's an RNA.
It's a ribosomal RNA. This is one of the examples in nature where RNA is catalytic. Okay, then the last stage is termination, the blue sock. And when termination happens, it is because we have arrived at a stop codon. And when there's a stop codon in the A site, there are no tRNAs that can bind it, right? If a tRNA had a binding anticodon for a stop codon, that would be a problem. But there isn't one. Instead, there are proteins called release factors that mimic tRNA's shape and structure. So if the ribosome has a spot that only a tRNA would normally bind, this release factor protein imitates a tRNA and slips in there and binds to the stop codon, okay? And when it does that, it doesn't show it in this picture, but there's energy involved. When it binds, it causes the release of the protein that's being made. It causes release of the tRNA that was previously attached to that protein, and it causes the large and small subunit to release. Okay, so the ribosome release factor is the curtain call for that ribosome. Once it's there, poof, everything dissipates. Okay, anything else? Oh, good. Well, that's good. That went quicker than I thought. Okay, any questions about that before we look at our last of the 3 sections and the most interesting, in my opinion?
What are you drinking? What are you drinking?
Whiskey. Half cut sweet tea. That's what I'm drinking.
Would have been more interesting if it would have been the first one.
Well, we'll never know. Okay, there's our guy Mendel. All right, let me look through our list here. Punnett squares for 1 and 2 genes, examples used in class, terms, blah, 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 pedigrees, blah, 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 inheritance and blood. Okay. All right, let's take a look. So, a little bit about the Punnett squares. Of course, we know this is a way to describe what Mendel had observed in his pea plants. So, Mendel's work was in the 1860s. Punnett squares came around like 1905, 1910, something like that. So there was a little bit of a stretch between, but once people started thinking about genetics again and revisited his work, rediscovered his work, I should say, they needed a way to organize those thoughts. And so Reginald Punnett was a geneticist, one of the first, and he invented Punnett squares. So before we do Punnett squares or discuss Punnett squares, for which we had our last quiz, let's think about a couple other terms, such as the parental and first and second filial generation. So we want to be comfortable if somebody is doing like an experimental cross between 2 organisms, we would call that the parental generation. That's the first 2 that are being crossed. Whatever offspring the parents produce, so if we have 2 parental organisms, they would produce offspring which are known as F1s. That's the first filial generation. If F1s were to produce offspring, those guys produce F2s. If F2s produce offspring, guess what's produced? etc. Okay, so each generation has a name, the parental being the first. Okay, if those organisms have genotypes, which is the alleles possessed by those organisms, so in this case, our parental generation had one that was homozygous dominant, which means it had two of the same. One was homozygous recessive, two of the same. They can only produce one type of offspring, which would be a heterozygote, which is one of each, right? And we know this because our Punnett square tells us so. Fortunately, you've had lots of practice with this in your lab. And we pair it up like that. We put the alleles from one organism in the vertical, alleles from the other on the horizontal, and then magic happens. Okay? Now, the second generation, this was Mendel's the so-called monohybrid cross, and this always produces the predictable ratio of 3 to 1. The 3 being the dominant phenotype, right? Phenotype being the physical expression of the genotype, and the 1 being the recessive. So 3 to 1 ratio. So make sure you look back over those concepts, the quiz that we did, and so forth. All right, if we do a double cross, let's see, do we have one example with, here we go. Make sure that you're comfortable with this. There might be a question or two about that. It's a skill you will need in future courses if you continue on in science. And the main thing to remember, I'm not going to spell it all out, I'm just going to do one little part of it. The main thing to remember is that when you produce gametes from a parental cross like this, all right, the gametes are what are going to be on the vertical and the horizontal axis, right? So keep in mind that if there are two genes, the gametes have to include a copy from each. So you'd have to have big Y and big R, or you could have big Y and little r. We don't do YY and RR. They have to have one copy of each. The little y, in turn, could also match up with either the big R or the little r. So that's one axis. And then on the top, again, I won't go through the whole thing, but I'm just going to set it up for you. On the top, we should have another 4 set of gametes. Now, in this case, they'll all be the same, but just as the exercise, remember, this Y could go with both R's and the other Y could go with both R's. It just turns out in this example, they're all the same. All right, then you can pair them up and you can see what the phenotype would be, what they would come up with based on whether you have dominant or recessive. If the parents were both heterozygous for each allele, then you would get a 9 to 3 to 3 to 1 every time, just like you get a 3 to 1 with this every time. But if you mix it up a little, and one of the parents is not heterozygous for each of the dominant and recessive alleles, then you'll get something different. In this case, it would end up being, well, actually if we do the first column, all the other ones are the same, right? These would all be the same. Those would all be the same. Those would all be the same. So you would get a 1 to 1 to 1 to 1 ratio, where 4 of the possible outcomes would be dominant for both, 4 of the possible outcomes would be dominant for Y, 4 dominant for R, 4 recessive for both. So a big change from 9 to 3 to 3 to 1. So it's good to work it out. All right, then we did some examples. One of the examples, I guess, hey, where are my arrows? I thought we had arrows that crossed. I guess I drew them on. One of the examples had to do with a very commonly mutated gene in organisms, which is the MC1R gene. And the MC1R gene, how do I describe it on there? Oh yeah, the E locus. The MC1R gene is usually designated as an E. So if you had a dominant copy, at least one, or homozygous dominant, in most organisms this would result in a very dark color, like a black color. If you had a recessive, and this is specific for our cattle example here, you would have a brown or some people call that red. And so you could do some crosses, like the crosses that we have up there, and make some predictions if you had the red cow on the left, and you had the, well, I can make a Punnett square of it, yeah. And the black bull on the right. Oh no, the black bull's in the middle, that's right, we're crossing horizontally. You could just set up a quick Punnett square if you want. This one you could probably do in your head, but why? Take the chance. So everyone is going to end up with the dominant E, which means everyone will be black. All the offspring will be black. Now this is complicated slightly by the presence of the white face. And the white face, what was the third gene? Oh, TERP1. The white face is encoded by a mutation in the gene called KIT. And so in the case of those cows that have a white face, or the bulls, or steers, whichever you're talking about, these guys would have a dominant mutation. If you were recessive, you as an animal would be solid colored, whatever that color was, black or red. Okay? So we could make a prediction like this guy over here on the far left, or this gal, I should say. She has at least one copy, actually she would have two copies of the dominant if she had that much white. And if she produced offspring with this gentleman in the middle, who was solid colored, then her offspring would be heterozygous. And in the case of cattle, heterozygosity for the KIT gene gives you the cute little baldy. Love those guys. What's not to love? And then the other example, it said, the bee locust in dogs, TERP1. This is when you have, so for dogs, it's a little bit different. This is for cows, or cattle. For dogs, the difference is that if you're recessive for that mutation in the MC1R gene, you are going to be yellow. If you are dominant, you're going to be brown or black. And so the question becomes, how do you determine whether it's brown or black? And that has to do with gene number 3 of the examples I gave you, which is TERP1. And the TERP1 gene, well, I'll just use the actual alleles. If you have a dominant copy of that, you'll be black. And if you have a recessive homozygous state, you will be chocolate or brown. So you could look at an example of an animal that had like this, and what color would they be? Remember, this is also an example, sorry, forgot. This is also an example of epistasis. That should be on there, is that on there? Actually, it isn't on there, is it? Well, it is an example of epistasis where two genes affect each other. So you have the ability to make black pigment, but because you're homozygous recessive for the MC1R, that overrides the other one, and this ends up being a yellow lab, right? If you are able to put pigment into your fur, but you have homozygous recessivity, for the TERP1 gene, then you're going to be a chocolate lab. Cute little guys. And then the other possibility, let's erase our cattle. The other possibility would be that you could express pigment in your fur, and you'd have at least one copy of the big B, and these would be your black labs. So imagine breeding these together, you get all kinds of wild possible outcomes. Well, not wild, all kinds of domestic dog outcomes. Okay, terms associated, dominant and recessive, we've been talking about those extensively. Heterozygous, two different. Codominant and incomplete dominant. Let's see, we have, how did we even do that? Where are the codominant and dominant? Here we go. So for codominant and dominant and incomplete dominance, that's where we have some examples like incomplete dominance. is where you have a blending of alleles. So the easiest way to think of this sometimes is in color-causing phenotypes, or genotypes, excuse me. So if you have a red and white, and then the combination of the two is pink, one is not overriding the other, right? They're both there. So you would call that incomplete dominance. Codominance is a little bit trickier. Codominance is when two things are there at the same time, right? It's hard to imagine sometimes because like it's hard to think of a lot of examples where two things can coexist as equals. But the preeminent example, which I think, didn't you do some of these on your lab exams, the blood types?
Yeah, there was one question that wanted you to figure out like Darth Vader and all them.
Oh yeah, I remember that. That's an old question. And so the difference here is that you have both dominance and codominance. So for example, if you're talking about little I, which is would cause the O type, these guys are just plain recessive. So that's our type O is a better way to say it. But if you have either A or B, they are just plainly dominant over that little I. The codominance comes when they're joined. So if you have A and B together, you're neither A or B, you're both. You're type AB. And so again, lots of different combinations you could do with Punnett squares with those guys as well. All right, okay, so I guess the last thing is, we didn't do Hardy-Weinberg, don't worry about that. The last thing would be the pedigrees, and there aren't going to be a ton of pedigrees. So I'm just going to ask you a few of these. This test is not going to be any longer than your other ones. So let's take a look at a dominant and recessive pedigree, and then we'll wrap things up here. So for pedigrees, there are going to be 4 possibilities. The two most straightforward would be autosomal dominant and autosomal recessive. Remember, this means it's a trait that is not on the sex chromosome. So if it's autosomal dominant, what we would expect, I guess we could use this to save chalk. What we would expect is that it's in every generation. So in other words, if we had an individual that had a dominant trait, and in a pedigree, having the trait means that you have a filled in square or circle. If you're square, you're a male. Circle, you're a female. So having the dominant trait means you had a parent that had it. Your parent had a parent that had it, and so on. So wherever there's an individual that has it, there should be a parent above them that also has it. Here, here. Okay, the fact that they don't have it down here doesn't make it not dominant. It just means this guy didn't happen to pass on his allele to his offspring. But it's still dominant because he had a parent that had it. Autosomal recessive, on the other hand, if we look at autosomal recessive traits, they are able to skip generations. Now, I won't draw a pedigree that has these half-marked things. So if you see a pedigree like that on one of my exams or one of my quizzes, it's always going to look, I'm drawing the same one that's up there, but it's going to look like this, because it's easier to understand. So if you don't draw the carriers in, you can really see the pattern better, which is that these individuals in the last generation have a trait that their parents didn't have. That's what it means to skip a generation. This individual has a trait that his parents didn't have, right? This can't be dominant. It would have to be in every unbroken line of descent. But in this case, there are individuals that have a trait and their parent doesn't. An individual has a trait and their parents don't. Okay? Now, X-linked recessive gets a little more complicated. I don't know that chart. We did not talk about the cat. I had a question on the test about this cat. We didn't talk about it, so I'm going to have to give you the answer to that question because it's actually a pretty long topic. But in any case, with X-linked recessive and X-linked dominant, the main difference in X-linked recessive, if we had a mom that was a carrier for the trait, let's say she has 2 X chromosomes, I'm going to put a little R there to show that one of her X chromosomes has a recessive trait. She can give that recessive trait, or that X, to half of her sons. If she does that, he will show the trait, right? This is the deal with X chromosomes. A male only has one, and so he can't hide whatever trait he gets on his X chromosome. It doesn't matter if it's dominant or recessive in the female. For a male, he just has the one X. He can't hide his trait. And so that male would be affected. If she had a daughter, and she gave that daughter her recessive gene, but the father, who doesn't have it, or he'd be shaded in, gave her the regular X, she'll be a carrier. And she could have a son to whom she gives her unaffected X, and he would be unaffected. Okay, and then again, in the next generation, if this male passes it on, let's assume his mate is unaffected. If he passes it on, all of his daughters will get his X chromosome, but they'll still just be carriers. His sons will get his Y chromosome, so they can't be affected by his trait. So it tends to skip generations, and it is more common in males because they only need one X. Males can't be carriers. That's one of the defining characteristics, actually. Males cannot be carriers. And then the last one, I guess our last teaching moment of the semester. Sad. Is X-linked dominant. And X-linked dominant is really easy to spot if you happen to get this type of example, because if a male has the trait, I'll put a D there for, he has an X chromosome that has the trait, and let's say his partner doesn't. You may remember me drawing this one out. I won't be as exaggerative as I was the first time. But a male a male determines the sex of his offspring, right? Because the female will give everyone an X. She doesn't determine the sex, but the male will either give a Y to his sons or an X to his daughters. All of his daughters, if they get that X, will have the trait, right? It's dominant. So if they get one copy, they'll have it. So the distinctive pattern is that whenever, I forgot to give him the trait. Whenever a male has it, every daughter gets it, none of the sons. The sons cannot get it from their dad, and the daughters have to get it. So for X-linked dominant, the distinctive pattern is 100% daughters get it from an affected dad, but 0% of sons. A little awkward grammar, but I think you understand. Okay, so that's the pattern to look for. Drop the chalk.